Good day everyone, I am April Gindi Sahor and today I'm going to report the Serratia. Morphology. Serratia species are gram-negative rod-shaped bacteria. Staining reactions, gram-negative. Culture characteristics. Serratia bacteria are facultative anaerobes and can grow in a wide range of environments, including water, soil, and some plants. Some species may exhibit red pigmentation on certain agar media. Biochemical or physiologic behavior. The ideal temperature range of Serratia bacteria to flourish is normally between 30 and 37 Celsius, 86 and 98.6 Fahrenheit. Although they are able to endure a broad variety of pH values, they generally prefer slightly acidic to neutral environments, with a pH range of 6.5 to 7.5 being ideal. Although they are often favor aerobic settings, Serratia species are facultative anaerobes, meaning they can grow in both aerobic and anaerobic situations. Because they are not very salt tolerant, Serratia species usually thrive best on media with low to moderate salt concentrations. Genetic analysis involves studying an organism's DNA to understand its genetic makeup. In a DNA study of Serratia, genotype would include specific DNA sequences, while phenotype would encompass observable traits influenced by those sequences, potentially including changes due to mutations such as antibiotic resistance or altered metabolism. Immunologic differences. Serratia species may have specific surface antigens that can trigger immune responses. Understanding these differences can be important for diagnostic and therapeutic purposes. So the next reporter we have Ms. Segundo.
Good day everyone, my name is Carisha Segundo. I am tasked to report about Pseudomonas. The Pseudomonas morphology, gram-negative and rod-shaped bacteria. Staining reactions, gram-negative. Cultural characteristics, aerobic, widely distributed in soil and water environments. Biochemical or physiologic behavior. Some species are opportunistic pathogens that can cause infections in immunocompromised individuals. The genetics analysis. Genetic studies can be used to identify different species and antibiotic resistance mechanisms. Animal inculations. Pseudomonas species can be used to study infections in animal models. Immunologic differences. Different species or strain may have unique surface antigens that can trigger immune responses.
Good day everyone, I am Baby Rawan Salipawan and I'll be reporting Clostridium bacteria. Clostridium bacteria is a genus of gram-positive, rod-shaped bacteria that form spores. Here are some characteristics of Clostridium. Morphology. Clostridium bacteria are rod-shaped with rounded ends. They can appear singly or in pairs. Staining reaction. Clostridium bacteria are gram-positive, meaning they retain the crystal violet dye in the gram staining process. Cultural characteristic. Clostridium bacteria are anaerobic, meaning they can grow in the absence of oxygen. They typically form colonies that are round, smooth, and they may reduce gas. Biochemical or physiologic behavior. Clostridium species are known for their ability to produce endospores, which are resistant structures that allow them to survive harsh conditions. They are also capable of fermenting various substrates to produce a variety of metabolic byproducts. Genetic analysis. Clostridium species can involve studying both genotype and phenotype. Genotype analysis focuses on genetic makeup of organisms, which include DNA sequencing to identify specific genes and mutations. Phenotype analysis, on the other hand, involves studying the observable characteristic or traits of the organism, such as its growth, pattern, metabolic activities, and response to various environment conditions. Overall, a comprehensive study of Clostridium bacteria would involve both genotype and phenotype analysis, include DNA sequencing to identify mutation and their potential impact on the organism characteristic and behavior. Immunologic difference, different species and strain may have unique antigenic profiles that can be targeted for diagnosis or therapeutic.
Good afternoon everyone, I'm Marcelina Said. So now I'm going to report about actinomyces. Morphology. Actinomyces are filamentous bacteria. A staining reaction is gram-positive bacteria. Cultural characteristic. Actinomyces can grow on specific culture media designed for their cultivation. Biochemical and physiological behavior of actinomyces. Temperature. Actinomyces bacteria prefer moderate temperature for growth, typically around 37 degrees Celsius, which is similar to human body temperature. pH. Actinomyces bacteria thrive in slightly acidic to neutral pH environments, with a pH range of approximately 6.5 to 7.5. Salt content. Actinomyces are generally not halopelic bacteria, meaning they do not require high salt concentration for growth. They typically grow well in a low to moderate salt environments. Atmospheric conditions. Actinomyces are aerobes, meaning they require oxygen for growth. They grow best in the presence of atmospheric oxygen, are not adapted to aerobic conditions. Genetic analysis. Genetic information about actinomyces can include genotype, phenotype, presence of mutations, and DNA studies to understand its genetic makeup. Immunologic differences. Understanding the susceptibility of actinomyces to different types of infection and its interaction with the immune system can provide insight to its pathogenicity and immune responses. That's all. Thank you.
Good day everyone, I'm Eliza C. Palmes. So now I'm going to report the Escherichia coli or E. coli. Morphology, gram-negative, rod-shaped, bacteria commonly found in the gut. Staining reactions, gram-negative. Cultural characteristic, facultative anaerobes that can grow in various environments, including the intestine of the warm-blooded animals. Biochemical or physiologic behavior. Some strains are pathogenic and can cause illnesses like urinary tract infections and food poisoning. Genetic analysis. E. coli genomes can be sequenced to identify virulence factors and genetic relationships between strains. Animal inoculation used in research to study host pathogens. Immunologic differences. Strains of E. coli may have different surface antigens that can be targeted by the immune system.
So good evening. My name is Hannah D.K. Omar. So I'll be reporting Mycobacterium tuberculosis. So Mycobacterium tuberculosis, the causative agent of tuberculosis, is a rod-shaped, acid-fast aerobic bacterium. It stains acid-fast using the acid-fast staining method. Mycobacterium tuberculosis has unique cultural characteristics, requiring special culture condition and exhibiting a slow growth rate, which makes it challenging to isolate and identify. Furthermore, it has a complex cell wall with unique components like mycelic acid. Genetic analysis, including molecular techniques, are used to study the genetic of Mycobacterium tuberculosis, including drug-resistant mechanism and strain differentiation. Animal models are used to study tuberculosis pathogenesis drug efficacy, and vaccine development. Mycobacterium tuberculosis triggers specific immune responses in the host, and understanding this instruction is important for development diagnostics and vaccines.
Hello, good day everyone. Just a review in Mycobacterium tuberculosis. Morphology: Mycobacterium tuberculosis is a bacillus, which means it has a rod-shaped appearance under the microscope. Staining reaction: Mycobacterium tuberculosis is classified as acid-fast bacteria, which means it retains the primary stain even after being exposed to acid decolorized. This staining properly distinguishes it as acid-fast positive rather than being classified as gram-positive or negative. Cultural characteristics: Mycobacterium tuberculosis usually cultured on specific media like Lewis-stained ginseng, medium or Meadowbrook ZH10 agar. This media selective for mycobacteria and provide an environment suitable for their growth. Biochemical or psychological behavior: Temperature: Mycobacterium tuberculosis grows best at temperature around 37 degrees Celsius, which is similar to human body temperature. pH: Mycobacterium tuberculosis prefers slightly acidic conditions with an optimum pH range of 6.6 to 6.8. Salt content: Mycobacterium tuberculosis is not a halophilic bacterium and does not require high salt content for growth. Atmospheric condition: Mycobacterium tuberculosis is a facultative intracellular pathogens and can survive and replicate inside host macrophages. It can grow both aerobically and anaerobically. Genetic analysis: Genotype: Mycobacterium tuberculosis has complex genotype with different strains exhibiting various genetic variations. Phenotype: the expression of certain genes. Mycobacterium tuberculosis contributes to its virulence, drug resistance phenotype. Mutations in specific genes of Mycobacterium tuberculosis can lead to drug resistance impacting treatment. DNA studies such as whole genome sequencing are used to understand the genetic diversity within M or Mycobacterium tuberculosis populations track transmissions pattern. Immunologic differences: susceptibility to infection: individuals with compromised immune systems such as those with HIV or AIDS or malnutrition are more susceptible to developing active tuberculosis mycobacterium infections. Immune functions: Mycobacterium tuberculosis has developed mechanism to evade the host immune response, including surviving within macrophages and modulating immune signaling pathways. Understanding these immune evasion strategies is crucial for developing effective treatments and vaccines against tuberculosis. That's all for Mycobacterium tuberculosis.